Our next speaker is Nicholas Hammerman. Nicholas, can you please share your screen and turn on your microphone and your camera? Hi, Nick, how are you?
Good morning.
Good to see you.
Yourself?
I'm good, thanks. Nicholas is from the University of Queensland, and his talk is titled "The Response of Red Sea Coral Communities to Recent Disturbance Events Vary Along a Latitudinal Gradient." And just a quick reminder for the audience, if you have a question for Nick or one of our other speakers, please type it in using the Q&A box at the bottom of the screen. Thank you, Nicholas.
Cool. Thank you all very much for rejoining us this afternoon. Again, my name is Nicholas Hammerman, and today I'll be presenting a chapter of my PhD thesis entitled "The Response of Red Sea Coral Communities to Recent Disturbance Events Vary Along a Latitudinal Gradient." So, as I'm sure we're all acutely aware of, global warming is having profound consequences for the functioning of corals, and in particular can cause them to expel their intracellular zooxanthellae, causing bleaching and, if unabated, mortality. And we all know that warmer El Nino, thermal stress, and resulting bleaching events are predicted to become more severe and frequent in the future. However, the consequences of ocean warming for reefs vary across their latitudinal distribution. Latitudinal differences in species composition and spatial variation in environmental conditions can influence bleaching prevalence and recovery potential. However, the coral reefs of the Red Sea are interesting as they span a broad latitudinal distribution and are subject to well-defined gradients of temperature, nutrients, and salinity. Northern Red Sea reefs between 24 degrees north and 30 degrees north are some of the most northerly reef systems in the world and experience lower summer SSTs as compared to the southern Red Sea, which has led to the speculation of the region being a potential climate refuge for future coral reef development. In contrast, coral reefs in the central and southern Red Sea reach summer SSTs which far exceed the average for coral reefs of the Atlantic, Indian, and Pacific Oceans. And these thermal gradients have strongly influenced the severity of recent bleaching events. Indeed, in 1998, 2010, and 2015, bleaching have had severe impacts on population dynamics and resulted in declines in species richness in the Saudi Arabian Red Sea, resulting in shifts to more thermally tolerant genera, especially in the central and southern Red Sea. And compounding these impacts so that roughly 60% of all Red Sea reefs are affected by other local stressors, such as overfishing, eutrophication, coastal development, and crown of thorns starfish outbreaks. However, in the absence of long-term monitoring, comparisons of the modern living coral assemblages and adjacent dead coral assemblages can provide insight into recent changes in community structure, and particularly across this latitudinal distribution in the Red Sea. And this approach, combined with high-precision radiometric age dating of dead coral skeletons, can provide critical insight into the timing of coral mortality and recovery in response to disturbance. Thus, here, we use uranium-thorium dating and comparisons of live dead coral assemblages to explore the response of Saudi Arabian Red Sea coral communities to recent disturbance events. And we surveyed reefs throughout this latitudinal distribution to discern differences in response across a gradient of temperature and local stress. Thus, this approach allowed us to explore 1, changes in coral community structure, 2, the timing of mortality, and 3, whether response to disturbances varied along a latitudinal gradient in the Saudi Arabian Red Sea. In order to answer these questions, we surveyed these 4 regions of Yanbu, Thuwal, Al-Leith, and the Faris and Banks, and within each region, we surveyed 2 to 3 sites. And within each site, we surveyed 4 20-meter phototransects at 5-meter reef slope, and we gathered all of our data for both the live and the dead coral assemblages from 50 random overlaid points over top of each photo quadrat. And to constrain the timing of impacts to corals from acute disturbances in each of these 4 regions, we randomly sampled dead coral colonies still in growth position from Acropora and Pocillopora, and then solely Porites from Thuwal. And coral samples were taken from well-preserved areas near the growth margin. And by dating dead coral skeletal material that represents recent growth just prior to coral mortality, the U-series age distributions produced from multiple fragments closely approximate episodes of mortality. So to begin with our results, each region within this non-metric multidimensional scaling plot formed a distinct group. And please orient yourself. Each of those symbols represents a transect per region. The open triangles from Yanbu, the open circle from Thuwal, the closed square from Al-Leith, and the crosshair from the Faris and Banks. And these patterns are mostly driven by regional differences in the relative proportion of live coral, dead coral, soft coral, other substrates such as sand, pavement, and clams, and algae. And generally, live coral followed a north to south gradient with the highest relative abundance of coral in Yanbu and the lowest relative abundance of coral in the Farrison Banks. And also, as you can see with the histogram on the right, the regions of Thuwal, Al-Leith, and the Farrison Banks had dead coral which was significantly greater than the relative proportion of live coral. Next, we wanted to understand, okay, but what were the contributors to each of the live and dead coral assemblages per region? And we have another histogram here with the genus on the x-axis and relative abundance again on the y-axis. And what we can see for Yanbu is that Acropora and Porites were the largest contributors to the live assemblage, followed by Pocillopora and Montipora, whereas the dead assemblages were mainly comprised of Acropora and Porites. Next, for Thuwal, we see that Porites was the largest contributor to the live assemblages, followed by Goniostrea and Pocillopora, whereas the dead assemblages were mostly Acropora, followed by Porites. For Al-Leith, we see that Al-Leith had a near equal proportion of Pocillopora and Porites within the live coral assemblages, whereas the dead assemblages contained dead Porites in the highest relative abundance, with moderate contributions from Acropora, Pocillopora, Goniostrea, and Montipora. And lastly, for the Farrison Banks, which also had the lowest live coral, we see that live assemblages were primarily comprised of massive Porites, and the dead assemblages were characterized by Acropora, followed by Porites. Next, we constructed scatter plots to just provide a direct comparison of taxa across all sites, and were thus used to infer changes in dominant coral genus between the dead and live corals. So what we have here first is dead coral percent on the y-axis, live coral percent on the x-axis. And for all regions, we can see that they had a greater relative abundance of dead Acropora as opposed to live Acropora. Next, when we look at massive Porites, we can see that Yanbu had a similar proportion of live versus dead Porites, whereas Thuwal, Al-Leith, and the Farrison Banks had a greater proportion of live versus dead Porites. And lastly, for Pocillopora, which was the third most abundant coral overall, and please note the change in the x and the y-axis within the scatter plot, we can see again that Yanbu had a similar proportion of dead as opposed to live Pocillopora, whereas Thuwal and Al-Leith had a greater proportion of live versus dead Pocillopora, and then lastly, the Farrison Banks had a greater proportion of dead than live Pocillopora. And next, we wanted to ordinate these communities across both regions to just understand, well, what's driving also the differences in the live and the dead assemblages as a community. And what we can see for the live coral composition, again, is that there is actually a fair degree of overlap as dictated here with these 95% confidence ellipses, the dotted lines of each region between Yanbu, Thuwal, and Al-Leith. However, the Farrison Banks do form a rather distinct group. And these differences are mostly driven by the relative contribution of key genera such as Acropora, Pocillopora, Goniostrea, and Porites. And we ordinated a similar thing for the dead coral composition, and we can see the same genera. However, there's a much more homogeneous plotting going on here, and that's because all of the transects within each region mostly contain dead corals from these same genera. So next, we wanted to temporally constrain for when mortality occurred and look at subsequent recovery. So let me orient you first to these plots. As you can see, on the x-axis is time, and then on the y-axis represents density, or represents these kernel density estimates, which are the shaded regions, and they really represent the peak distribution of dates. They have a black outline for Pocillopora and a gray outline for Acropora. And then each date is depicted as either an open circle for Acropora or a black circle for Pocillopora. And I have here a square for Porites, but again, remember please that I'd only dated those from the next plot I'll show you in Thuwal. And each date is shown with its associated error bar. And lastly, the vertical dashed lines represent recorded bleaching events per region. And please note they're offset, as that takes into account the specific month and that year in which the bleaching occurred. And what we can see for Yanbu is that 86% of the branching dates for both Acropora and Pocillopora fell between 2006 and 2017, and roughly half of these dates were within 1 to 2 years of 2010. Next, for Thuwal, we can see that there's an added vertical dashed line for bleaching in 1998, as well as those Porites dates. And first, we see for the branching dates of Acropora and Pocillopora that 70% of them occur between 2010 and 2016, and 80% of them occur within plus or minus 2 years of 2015. And the 20 dead massive Porites occur over a much broader distribution from 1990 to 2015. Lastly, for Alith, we have a dotted line at 2010 because that's where we detected degree heating weeks, yet no literature reported bleaching. And we can see that Acropora was first had its peak in 2010, and then Pocillopora later in 2015. And lastly, for the Faris and Banks, we also now have a solid black line, which represents a COTS outbreak in 2009. And we had the largest distribution of dates within 2 years of 2010. So thus we found a latitudinal driven difference in coral community response to recent disturbance events, whereas reefs in the north of Yanbu had the coral composite or had a composition dominated by live hard corals, as well as the highest relative abundance of Acropora. Most of the mortality of both genera are mostly due to 2010 bleaching event, which is corroborated with the literature, as that event caused more severe effects for Yanbu as than the 2015 bleaching. And similarity between the coral live and dead assemblages, as well as the highest coral abundance, suggests either the lowest mortality rate during recent bleaching events or the quickest recovery. And next, for the more central Thual and Alith regions, we show that especially within Thual, there's a compositional change from a fast-growing reedy Acropora to a slower-growing and more thermally tolerant Porites goniastrella assemblage. And in Alith had a similar assemblage of Porites and Pocillopora as previous literature, but in both regions, the benthic structure was dominated by dead corals. And as we can see, the mortality of both branching genera were largely constrained to 2010 and 2015. But please note and remember for Thual, the broader distribution of Porites dates imply a more persistent community across time. And lastly, in the Faris and Banks, we observed the most pronounced reduction in coral cover from reef surveyed, a live coral composition dominated by Porites and no live Acroporids or Pocilloporids, and the comparison between the live and dead assemblages revealed a significant change in coral dominance from Acropora to Porites. And the mortality peak distribution occurred around 2010, most likely due to both a combination of a major COTS outbreak in that area, as well as thermal stress in 2010. So in conclusion, we show the 3 southerly regions have undergone a homogenization of coral community composition and decrease in coral cover. We show a decreasing trend in Acropora abundance moving south, similar to the Arabian and Persian Gulf. And we show that the northern coral reefs are able to recover and maintain composition following disturbances. However, we also show the increased vulnerability of central and particularly southern Red Sea coral reefs to further detriment from continuing disturbances. And with that, I thank you all very much for your attention, as well as my collaborators from the King Abdullah University of Science and Technology, as well as all my colleagues and co-authors from the Marine Paleoecology Lab and UQ in general. Thank you very much.